So we're going to start off this lecture by going over the mechanisms of how tubular reabsorption and tubular secretion happens. So throughout the nephron, we have seen that the lumen is lined by simple epithelium. There are parts with simple squamous epithelium, and then there are parts with microvillate simple cuboidal epithelium. So the things that are either getting secreted into the lumen of the nephron or are getting reabsorbed into the bloodstream take one of two modes of transport. The first is paracellular transportation. So where stuff is moving between epithelial cells. So the gap between two cells is where the substances are moving through. Transcellular transport is when the substances are actually moving through the epithelial cells itself. So here, you already know that the peritubular capillaries are the blood capillaries that are adjacent to the proximal and distal convoluted tubules. Now, the blood that's flowing in there has a fairly low hydrostatic pressure and a fairly high oncotic pressure. So this is more like your blood colloidal osmotic pressure, which is from the larger substances within the blood, such as the protein albumin. And most of the reabsorption in the PCT is happening because the surface area is high, since the cells lining the proximal convoluted tubule have microvilli. So if we look here, so let's get reoriented into this diagram first. So this is in the renal cortex right here, because we are looking at the length of the proximal convoluted tubule. Here is the glomerulus, and here is that capsular space that is lined with simple squamous epithelium. Now, these yellow dots coming out of the glomerulus are your filtrate. So filtrate enters your proximal convoluted tubule, and as you can see, this is lined with microvillated simple cuboidal epithelium. This is the lumen of the proximal convoluted tubule. Here is your peritubular blood capillary. So to recap, tubular reabsorption means that fluid or filtrate is going to go from this tube, go across, and get sucked into blood. That is tubular reabsorption. Tubular secretion is when stuff comes from blood, goes through the cell, and ends up in the filtrate tube. So the first mode of transport is when substances are moving between epithelial cells. So if you see these cell junctions right here, so they're moving in and out through the gaps between two cells here. So they're moving from these gaps between cells itself. That is paracellular transportation. Transcellular transportation is when substances have to, either by facilitated diffusion or transmembrane proteins, be included into this cell, your microvillated cuboidal cell, and then somehow exocytosed or by facilitated diffusion out of the cell into the the interstitial space, and then diffuse right into blood. So that is the difference between paracellular and transcellular transport. One of the things that is under normal circumstances, one of the things that is almost 100% reabsorbed into blood is a monosaccharide called glucose. Now here's what happens. This is the tubular fluid right here. If I change the color to black, let's orient ourselves here. So this is the lumen of a proximal convoluted tubule. So filtrate is passing through here in this direction. This is a very zoomed in version of your microvilliated simple cuboidal cells, and the microvilli here are functioning to increase surface area. Now, ideally, the filtrate, which has a lot of glucose in it, the glucose has to be reabsorbed back, because glucose is what your cell undergoes glycolysis, and that's the one that is going into the mitochondria for you to have Krebs cycle and ultimately generate ATP. So here's what's happening here. So the green dots here are the glucose molecule. The purple dots here are sodium. So in the tubular fluid, you are seeing a high concentration of sodium, but you are seeing a low concentration of glucose. Now here's what's happening. Sodium is moving from an area of high concentration into the cell. From the lumen of the proximal convoluted tubule, it is going through this transport protein, and it is diffusing inside this cuboidal cell, which is where there is a low sodium concentration. Now this is creating sort of a solvent drag, if you will. So sodium is moving from an area of high concentration to an area of low concentration through facilitated diffusion. However, as this is happening, through the same transport protein, you are seeing glucose being moved from an area of low concentration to an area of high concentration. So it's moving against or up its concentration gradient by secondary active transport. So this protein right here, this blue protein, that is the sodium glucose symporter. So what's happening here is wherever the sodium is going is where the glucose is going. Sodium happens to be going from an area of high concentration outside the cell to an area of low concentration inside the cell. Glucose, however, is going from an area of low concentration, it is going upstream, to an area of high concentration inside the cell. And so it's symported along with sodium. And this is secondary active transport. So glucose keeps being symported with sodium until there is about 0% glucose that remains in the filtrate or the tubular fluid. Now, once inside the cell, these glucose molecules are again using a transport protein. This time, it is a glucose uniporter. So this, because it's going from an area of high glucose concentration to an area of low glucose concentration, it is actually being transported via facilitated diffusion. So these glucose molecules are going to go through this transport protein. They're going to end up in this interstitial fluid. Then they're going to go by diffusion into this blood capillary and back into blood. So this is how glucose is reabsorbed back into the blood. And this is an example of transcellular transport, because glucose is not going between two cuboidal cells and into the interstitial fluid. It is having to pass through an individual cuboidal cell via transport proteins, go through the body of the cell and then get diffused out, and that's how it gets reabsorbed into blood. Now, glucose, however, does act as an osmotic diuretic. A diuretic is a substance that makes you lose water. So a diuretic is a substance that decreases water reabsorption in the nephron. So the less water you're reabsorbing into your body, the more water becomes urine. So what glucose does is that it pulls water into the tubular fluid, and it causes loss of fluid in urine because you're peeing out more since it acts as a diuretic. So this condition where glucose is excreted in urine is called glucosuria, and it is a classic symptom of diabetes. And because there's frequent urination, you're losing water, so your blood volume goes down. When your blood volume goes down, your brain recognizes the loss of blood volume and gives you the sensation of thirst. So you go and hydrate yourself, and that in turns bring your blood volume back up. So some of the classic symptoms of diabetes are frequent urination, presence of glucose in urine, and excessive thirst as well. The next substance we're going to look at is actually sodium. So sodium is reabsorbed at various places within the nephron tubules. So about 98 to 100% of sodium is reabsorbed from the tubular fluid, aka filtrate, off into the blood. So it happens along the entire length of the nephron tubule. So it happens in the proximal convoluted tubule, in the loop of Henle, as well as the distal convoluted tubule. But the majority of it happens in the proximal. Now, the point to note about the distal convoluted tubule is where the absorption of sodium is regulated by hormones. So as we get into the distal convoluted tubule, so before the distal convoluted tubule, in the PCT and the nephron loop, it is happening by diffusion and concentration gradient changes. In the distal convoluted tubule specifically, it is regulated by hormones, which we've already looked at in the RAAA system that we saw in the previous lecture, the RA system. And one of the hormones that we saw was aldosterone. So aldosterone makes you reabsorb sodium. It makes sodium go from your filtrate back into your blood. And it is synthesized by your adrenal glands. Now, wherever sodium goes, water tends to go there as well. So if sodium is the more sodium is reabsorbed into blood, think about what that would do to urine volume, right? So more sodium being reabsorbed would also result in more water being conserved. And more water being conserved means that you're peeing less. So aldosterone is not a diuretic. It is the opposite of a diuretic. It has a mild anti-diuretic effect because it helps you reabsorb sodium, and because it reabsorbs sodium, it absorbs water as well. And it typically happens when you have low blood pressure. We saw the entire RA AAA system get activated when there was loss of blood volume or low blood pressure. The opposite of that is when your blood pressure is too high. When your blood pressure is too high, your blood volume has increased, and your heart senses, the atria of your heart senses that, and it secretes something called atrial natriuretic. So Uretic, the Latin name of sodium is natrium, which is where the Na comes from. And so natriuretic peptide makes you pee out sodium. That is what a natriuretic means. So it pulls sodium into the filtrate, or it prevents reabsorption of sodium. And this hormone is synthesized by the atria, which are the two superior chambers of your heart. So with people, dietary intake of sodium or salt varies significantly, which is why if someone is running a low blood pressure, they're encouraged to have salty snacks, and that's how blood pressure comes up. And if someone has hypertension or high blood pressure, they're usually advised to eat less salt in general. Next, we're going to look at where and how much is water absorbed. And I'm going to explain that based on this diagram. Starting with the proximal convoluted tubule. So this purple segment here, about 65% of water is absorbed in the proximal convoluted tubule. And this is obligatory water reabsorption. So like out of the filtrate, approximately 65% is almost always reabsorbed here in the proximal convoluted tubule. Now, in the loop of Henle is about 10% of water gets absorbed, because remember, out of the filtrate, 99% of the filtrate gets reabsorbed, and the filtrate is mostly water. Now, when you get to the collecting duct, this is when your blood pressure and your blood volume determines how concentrated or how dilute your urine gets. So this is where regulated water reabsorption happens. So if you're dehydrated, your body wants to conserve water, and this is where a lot of water will get reabsorbed to make your urine concentrated. If you are hydrated well enough or hydrated in excess, this is where water gets added or it's secreted into the collecting duct. So you're peeing more, but your urine is dilute in that case. The next important ion is potassium ion. And we know potassium ion is important because we've seen it, muscles need potassium to come back to a relaxed state. Potassium ions are important for repolarization in general because of your sodium and potassium pump. So we've gone through the physiology of that already. So most of the potassium, again, is absorbed in the proximal convoluted tubule, about 60 to 80%, 10 to 20% in the nephron loop, and the rest is regulated in your collecting tubules and the collecting duct. Now, there are cells called intercalated cells that gauge the amount of potassium that is coming through the fluid here, and they reabsorb potassium here, the intercalated cells. Principal cells of the collecting duct, they vary in potassium secretion depending on what your aldosterone level is. So point to note here is most of the potassium is absorbed in the proximal convoluted tubule, about 20% in the nephron loop, and then in the collecting duct, the intercalated discs are reabsorbing your potassium continuously, and when there is excess potassium, your principal cells will decide if there is excess potassium, and those are the ones that will end up secreting potassium into your urine. So intercalated cells and principal cells are doing opposite things when it comes to potassium. The intercalated cells are reabsorbing it back into your bloodstream, versus your principal cells are secreting it into your urine. The next ions that we're looking at are the calcium ion and the phosphate ion. And we've already seen calcium ion and talked a little bit about blood calcium levels back in your bones unit. We did talk about how we maintain a homeostasis of blood calcium. When blood calcium goes below a certain range, it is called hypocalcemia. When it goes above a certain range, it's called hypercalcemia. Hypercalcemia relaxes muscles. Hypocalcemia makes them tense. So PTH here actually stands for parathyroid hormone. So absorption of or reabsorption of calcium and phosphates is largely determined by parathyroid hormone. Now here's what happens. First I'm going to talk about calcium and then I'll talk about the phosphate. So parathyroid hormone, which is secreted by your parathyroid glands, which are small nodular glands located on the posterior side of your thyroid gland. Now they target the distal convoluted tubule. And if you are hypocalcemic, meaning you don't have enough calcium in your bloodstream or you're not getting enough calcium through your diet, this parathyroid hormone is going to target your distal convoluted tubule, and it is going to have taken all this calcium ions back into your bloodstream. So it helps your body reabsorb, meaning take out calcium out of the filtrate and soon to be urine and take it back into your bloodstream so that you're not becoming even more hypocalcemic. So parathyroid hormone stimulates calcium reabsorption in the distal convoluted tubule. Now phosphate ions is a little bit different. There's an inversely proportional sort of relationship between what PTH does for calcium and what PTH does for phosphate. So parathyroid hormone inhibits phosphate reabsorption in the proximal convoluted tubule. So it's making you pee out excess phosphates by inhibiting the cells to pump them back into your bloodstream. As a result, you're going to pee out more phosphate because phosphate is not reabsorbed here and calcium is reabsorbed here. So you're going to pee less calcium out, but you're going to pee more phosphate out. Finally, we have the bicarbonate, which is HCO3 minus, and the hydrogen ion movement. So for the bicarbonate, almost all of it, about 90% of it, is reclaimed here in the proximal convoluted tubule. The remainder is reclaimed in the nephron loop. Now, once you get to the distal convoluted tubule, it is sort of regulated. So depending on the acidity or the alkalinity at the moment, here's where like it's decided whether phosphate and hydrogen ions are either reabsorbed or secreted. That depends on what the acid profile or the pH of the filtrate is at that point. So if your urine is too alkaline, HCO3 is reabsorbed, making it more acidic. If your urine is too acidic, then H plus is reabsorbed, making it less acidic, if that makes sense. Lastly, we are going to take a quick look at what are the products that are excreted as wastes, right? So nitrogenous wastes are mostly from breakdown of proteins and nucleic acids. So when we say there are nitrogenous wastes, there are wastes that come from metabolism. Urea is produced. Urea is a nitrogenous waste, and it's produced from protein breakdown or protein digestion. Uric acid is from breaking down nucleic acid. And depending on what the blood levels are, uric acid can be either reabsorbed or it can be secreted and eventually peed out. The next nitrogenous waste actually comes from muscle metabolism. And we talked about the creatine phosphate system when we did muscle metabolism in ANP1. But creatinine is produced from muscle metabolism, so muscle use, essentially. And this is secreted, and this is what you eventually end up peeing out. So creatinine does not get reabsorbed into the body. It's peed out. Additional waste products include drugs and other bioactive substances, sort of antibiotics, ibuprofen, aspirin, Tylenol, things like that. Anything you're taking in terms of pharmaceutical drugs, leftover metabolic wastes from hormones, bile products, things like that. Also, anything that's excess or deemed a waste product is also secreted into urine and then peed out. So I'm going to stop here, but please note that the last PowerPoint is self-study. The clinical terminology PowerPoint for urinary system is self-study and is available on Canvas. So please don't forget to go over that PowerPoint. There's going to be no video for that. You will be going over that on your own.